Hello students, today we are going to see syllabus for TYBSc paper 3. I am Dr. Darshana Subhash Korgaonkar, Assistant Professor, Department of Zoology, and this semester I am going to deal with your paper 3. So, the title for your paper is Genetics and Bioinformatics. So, your paper 3 comprises of 4 units. First unit is molecular biology, second is the genetic engineering, third one is human genetics, and fourth is the bioinformatics. So in first unit, that is the molecular biology, you're going to study different chemical and molecular processes that affect the genetic material. Also, you're going to study the concept of DNA damage and repair and how gene control is necessary for cell survival. So, in first unit, molecular biology, in first chapter, you're going to study different types of mutation. As you can see on screen, there are number of mutations listed. Also, you're going to study the trinucleotide repeat expansions and what are the conditions caused due to trinucleotide repeat expansions like Fragile X syndrome and Huntington disease. Also, you're going to study spontaneous mutations in which you will study tautomeric shifts and spontaneous lesions. In next chapter, you're going to study induced mutation. How the mutations are induced. Mutations can be natural, mutations can be induced. So there are number of reagents or number of substances which can be used to induce mutation in an particular DNA. So here you can see physical agents which are used to induce the mutations. Agents like X-rays, alpha rays, beta rays, gamma rays, UV radiations, these all are the physical agents which can induce a mutation in an organism. Next topic, you are going to study different chemical agents which can be used to induce mutation, like base analogues, intercalating agents, then deaminating agents, hydroxylating agents, alkylating agents, and alpha toxins. Next chapter, you are going to study the preventive and repair mechanisms for DNA damage. In this chapter, you are going to study how different enzymes work to repair the damaged DNA. In this chapter, you are also going to study the mechanisms like photoreactivation, excision repair, also some post replicational repair mechanisms like recombinational repair, mismatch repair, SOS repair, etc. In next chapter, you are going to study the eukaryotic gene expression. In that, you are going to study the regulatory protein domains like zinc fingers, Helicton's helix domain, and leucine zipper. Next unit is genetic engineering. This unit is included in your syllabus so that a set of techniques to modify an organism's genome to produce improved or novel genes and organisms is introduced to you all. In this unit, in first topic, you are going to study different tools in genetic engineering. That is, what are the basic requirements uh, what are the basic requirements as far as the genetic engineering is concerned. A basic knowledge is given through through this topic, like which are the enzymes, how the restriction enzymes are named, that is the nomenclature of restriction enzymes, what are the different types of restriction enzymes, which enzymes else are used in genetic engineering, etc. In next chapter, you're going to study the vectors, because vector is one of the most essential component as far as the recombination is concerned or genetic engineering is concerned. So there are many vectors developed. So what are these vectors? What are its general properties? What are the advantages and disadvantages? And what are the different vectors which are used in genetic engineering? Next, in next chapter, we are going to study the cloning techniques. In this, how cloning or how a recombinant DNA is prepared. Uh, we are going to see that in detail in this chapter. Then also along with that, we are going to see about the reverse transcriptase enzyme. Along with that, how genomic and cDNA libraries are um, maintained. We are also going to study that in this chapter. Next, we are also going to study the PCR techniques. PCR is a polymerase chain reaction and its applications. In next chapter, we are going to study the sequencing techniques. DNA sequencing is one of the major uh, research done or major research done in a genetic engineering field. Many scientists like Maxim and Gilbert Sanger have developed a technique for sequencing this DNA. So you're going to study this sequencing techniques in this chapter. Next, you're going to study the detection techniques like southern blotting techniques, that is southern blotting, northern blotting, western blotting, and the application of these blotting techniques. Third unit is human genetics. In this through this unit, you are going to be introduced with genetic alterations in human genome and their diagnosis. So in this topic, you are going to study the non-disjunction during mitosis and meiosis. So during when there is a non-disjunction during mitosis and meiosis, certain conditions are caused due to the unequal shifting of chromosomes, maybe. So there are number of alterations caused, and they may have some adverse effect, or they may also have some mild effects on a organism. So you are going to study in this first topic. Along with that, an aneuploidy, polyploidy conditions are going to be studied in this topic. Next is the genetic disorders. There are some disorders which are associated with the genes of an organism. So, if there is a defective gene, then if there is a defective gene in an organism, it may cause some disorder, or if the defective or if the mutation, or if the defective gene has not does not have any important role in an organism, it can be tolerated. But some diseases are caused because of this genetic disorder, that is there is a defect in gene by birth. So such disorders are also called as inborn errors of metabolism. We are going to study phenylketonuria, G6PD deficiency, alkaptonuria, albinism, etc. Next topic, we are going to see the diagnosis, how different types of prenatal diagnosis can be done, so that the parents beforehand may understand which disorders or diseases the child can face. So we are going to see that in this chapter. Next unit is bioinformatics. So a bioinformatics is included in your syllabus, so that a computational approach to learning the structure and organization of genomes, phylogeny, and metabolism can be introduced to the students. So in first chapter, there is an introduction to bioinformatics, then different bioinformatics web resources like NCBI, EBI, OMIM, PubMed, etc. Also, you are going to see the applications of bioinformatics. Next, you're also going to see the tools, databases, tools and their uses, like primary sequence databases, secondary sequence databases, derived databases, structured databases, etc. In next chapter, you're going to study sequence alignment methods like BLAST, FASTA, then pairwise and multiple sequence alignment, and significance of this sequence alignment. In next topic, you're going to study the predictive applications using DNA and protein sequences. In this chapter, you're going to study pharmacogenomics, metabolomics, then protein chips and functional proteomics, etc. So, this is your syllabus for paper 3, that is the genetics and bioinformatics for your semester 6. Thank you. Stay home, stay safe, keep learning.